The Watson and Crick model DNA is double helix. Now in 1951, James Watson learnt about X-ray diffraction pattern projected by DNA. The knowledge of the chemical structure of nucleotides is discovered by James Watson. That is, the DNA is composed of deoxyribose sugar, phosphate and nitrogenous bases. Erwin Chargaff experiment demonstrated that the ratio of A and T is 1:1 and G and C is also 1:1. That is, A plus T is equal to G plus C. Okay. In 1953, Watson and Crick proposed their double helical structure of DNA. Then Rosalind Franklin and Maurice Wilkins, they produced X-ray diffraction pattern by the DNA fibers. Then this is the double helical structure of DNA discovered by Watson and Crick, proposed by Watson and Crick. DNA is double helical, strands are anti-parallel with a backbone made up of sugar and phosphate. Here you can see sugar, phosphate residues are alternate. So the backbone of the DNA is made up of sugar and phosphate. Two strands, they run anti-parallel. Two strands wrap around each other every 30 angstrom and once every 10 base pair. Two chains are held together with the hydrogen bond between AT and GC. Now here you can see in this structure sugar phosphate backbone. These blue colored balls which you can see, these are sugar and phosphate molecules. This forms the backbone of the DNA, and the bases, they are embedded inside of the helix. Okay, bases are embedded inside of the helix. Then the DNA chemical constituents. DNA and RNA nucleotides. Now this is DNA nucleotide, which is composed of base, sugar and phosphate group. Okay, base, sugar and phosphate. Sugar present is deoxyribose sugar. Here at the 2' oxygen, it's absent. This is RNA nucleotide, which includes base, sugar, ribose sugar and phosphate. Base, ribose sugar and phosphate. Okay, this is nucleotide. Nucleotide means consists base, sugar and phosphate. This one is polynucleotide chain of the DNA molecule, where 5th carbon of the sugar is linked to the phosphate group. This is the 5th carbon of the sugar is linked to the phosphate group. Then again 3rd 3' OH of the sugar is linked to the next nucleotide's phosphate group. 3' end, see this one, 3' end is known as growing end of the DNA, because all the new nucleotides, or say nucleotides which are in one chain, they are linked together with the previous bases 3' OH. Okay, next base whatever is added into the same chain is linked to this free 3' OH. with the phosphate group of next nucleotide, which is now going to bind to the DNA molecule. Structurally, purines, that is adenine guanine, best pair with the pyrimidines. See this one, purine best pair with the pyrimidine. Pyrimidine if joins together, this is not feasible angle, the angle is very less. When purine joins, angle is high. So this is best pairing of purine and pyrimidine. Diameter is 20 angstrom. Always adenine pairs with the thymine and guanine pairs with the cytosine. Okay, adenine pairs with the thymine and guanine pairs with the cytosine. So this explains Chargaff's rule, why A is equal to T and G is equal to C in DNA molecule. A and T or G and C both pair with one another with the hydrogen bonds. There are 3 hydrogen bonds present between the G and C molecules and 2 present between A and T. 2 hydrogen bonds between A and T and 3 between A and G. See here also, adenine and thymine base pairing is shown, which consists 2 hydrogen bonds. Guanine and cytosine, they have 3 hydrogen bonds. Now here you can see the molecular structure of the DNA. These are 2 strands of the DNA which are coiled around one another. Each turn consists 10 base pair. Now see here, this angle of rotation is higher, so it is major groove. Here this angle of rotation is lower, so this is minor groove. Each turn of this helix consists 10 base pair and distance between 2 base pair. Now here you can see AT and GC base pair. The distance between 2 base pair adjacent nucleotides is 0.34 nanometer. Okay, so therefore one turn distance is 3.4 nanometer. Diameter of the DNA is, we have already seen, it is 2 nanometers or 20 angstrom. This is the axis around which the 2 DNA strands are turned around one another. This is B form of DNA, which is present in 99% of the organism, normal form of the DNA, okay, which is left-handed. This is right-handed DNA, okay. Now what do you mean by this left-handed and right-handed helix? I will show you in video, another video. This third one is Z form of DNA, which is zigzag DNA, found in only few archaeal organisms. The next experiment is Meselson-Stahl experiment. These two scientists, Meselson and Stahl, confirmed that DNA replicates by semi-conservative mode of replication. Okay. Semi-conservative mode, that is only 50% of the DNA strand is identical or is passed to the next generation and 50% is newly synthesized. So what they did is, for control, they took E. coli and grown for many generations in normal nitrogen, N14 nitrogen medium. Okay. Now why only nitrogen is considered here? Because DNA is made up of nitrogenous bases and they are synthesized by every organism whenever required or whenever DNA duplicates. Then they extracted the DNA from the cells, centrifuged on density gradient centrifugation. Now this density gradient centrifugation separates molecules on the basis of their density. Now if you see this tube, centrifuge tube, here the density gradient is already developed using either sucrose or ficol or cesium chloride. Density at bottom is highest and decreases towards the top side. So at the top side the density of the medium is lowest. Now this N14 nitrogen has low density. So it's all the DNA molecules extracted from this control, they are settled at the top side. Okay. Then E. coli grown for many generations in N15 medium. Same E. coli grown here is transferred, inoculum is transferred to the medium containing heavy nitrogen. N15 is heavy nitrogen. Then they extracted the DNA from the cells, allowed to centrifuge. They found that this DNA molecule settles at the bottom because whole DNA molecules contain N15 heavy nitrogen. Then N15 cells are transferred to N14 medium. Okay. From here they are transferred to here in the flask containing N14 normal nitrogen. The cells replicate once to produce first generation of the daughter cells. Then the DNA is extracted from such cells, centrifuged. Now since these organisms they have been grown first on heavy nitrogen and now they have grown on normal nitrogen, they will have one strand which is of light blue color and one strand with a pink color. Why? Because now the DNA present in a cell replicates two DNA strands. They separate from one another and this pink one is newly synthesized. So it is one DNA molecule contain one DNA strand with a heavy nitrogen and another one with the light nitrogen. Next, after 30 minutes or after growth of first generation, the cells replicate in second time to produce second generation of the daughter cell. DNA is extracted from the cells again, centrifuged, DNA is centrifuged onto the density gradient. Now here you will find two bands in the centrifuge tube, one with the light nitrogen and one with the mixed nitrogen strands, one heavy and one light. Now see this one, as these organisms grow, the cells having this type of DNA, one with the light nitrogen and one with the heavy nitrogen. In the next generation, these two strands will separate, and since now the cells are growing in light nitrogen, here one strand is fully pink because one parental strand is pink and newly synthesized strand is pink because now we are growing cells on N14 nitrogen, and these are the old strands, one with the blue color and one is newly synthesized. Okay. So using this experiment Meselson and Stahl discovered that the DNA replicates by semi-conservative mode of replication.